drugs, or ideas, what you consume influences who you become. On the Mind and Matter podcast, we learn together from the best scientists and thinkers alive today about how your mind-body reacts to what you feed it. Before starting Mind and Matter, I spent 10 years in academia doing scientific research. I got a Ph.D. in neuroscience where I focused on neuroendocrinology and the neurobiology of behavior, and before that, I specialized in molecular, developmental, and evolutionary genetics. I use my scientific background to help parse and translate the information that guests share on the podcast. In addition to the podcast, I write long-form written content inspired by the show where I integrate what I've learned across episodes. I also have a free weekly newsletter where I provide you with upcoming guests, share links, and provide commentary on scientific studies and research that I'm reading, and more. Visit mindandmatter.substack.com to find all of my content. This episode is ad-free, and I rely on my audience to support my efforts. If you're getting value from this content, please consider becoming a paid subscriber to the Mind and Matter Substack. For just $5 a month, you'll get early access to episodes and other content, full episode transcripts, and I'll prioritize answering your questions in emails and the comments sections. You can also support me by checking out the links in the episode description to my affiliate partners who make various health and wellness products that I use and enjoy myself. You'll receive a discount and help support the podcast that way. And if you want the benefits of a paid subscription, but it's not in your budget, simply sign up for my free weekly newsletter, send me an email, and I'll give you a complimentary paid subscription. Word of mouth is the best way to help the podcast grow. So if you like what I'm doing, please share your favorite episodes with family and friends. The purpose of the podcast is to provide you with information obtained largely from primary sources, the people doing the research and producing new knowledge. This content is never meant to serve as medical advice. And with that, enjoy the episode. Can you tell everyone a little bit about yourself, who you are, what your lab studies?
So I'm Greg Scherrer. I'm a professor at the University of North Carolina. In terms of background, I started in France, in the east part of France, at the University of Strasbourg. I started with a PharmD, then I did a Ph.D. So my background at the time was in opioid receptor biology. My supervisor during my Ph.D. was Brigitte Kiefer. And Brigitte is known as the person who first cloned the genes that encode opioid receptors, and then showed that one of these receptors, which is known as the mu-opioid receptor, is the one that's responsible both for the analgesic properties of opioids, so why we use them in the clinic, but also the side effects, including addiction. And so after this background, so when you sort of start with opioids, you can go different places. You can go more towards addiction or towards better understanding pain and opioid analgesia, and that's what I decided to do. So I moved to the US 2006, and I did 2 postdocs. My first postdoc was with Alan Basbaum at UCSF on neurobiology of pain, and after 3 years, I had a background at that time that was really pharmacology and molecular biology. So I had never taken a formal neuroscience class at that time. So I wanted to really become more a neurophysiologist. So I went to Columbia, spent a couple of years with Amy McDermott, where I learned spinal cord electrophysiology, and then started my first lab at Stanford in 2012. And recently I moved my lab to UNC.
So you're a pharmacologist who sort of became a neuroscientist, and that happened sort of via the study of pain and reward systems in the brain.
Exactly. Yeah.
So I want to talk about opioids a little bit. Most people know what they are, at least on one level. Obviously, these have been widely used medications. They are given as pain relievers, but of course, they can also be highly addictive. And it sounds like those two things are intimately related. So can you tell us a little bit more about the opioid system? You mentioned that there's this mu-opioid receptor that's responsible for both the analgesia, the pain relief, and the addictive properties of opioids. Are there other opioid receptors? Are these two things sort of, are they two sides of the same coin, or can you separate the addictive and the pain relieving parts of this?
Great question. So, the first studies that really demonstrated that there were sites in the brain that were binding opioids, such as morphine, fentanyl, oxycodone, all these things sort of work the same way, suggested that there are specific proteins that neurons express that can bind these drugs. And so it's really the discovery of the genes that sort of clarified what the endogenous opioid system is. So our body expresses a number of receptors and a number of peptides that are part of what we call the endogenous opioid system. So this is a system, people don't always fully realize this, but essentially what these very dangerous drugs do is to hijack our endogenous opioid system. But normally in our body, even when things function perfectly well, our cells can release some endogenous opioids that are known as endorphins or enkephalins that produce effects that are useful for us, that are not dangerous. And they do that by acting on these receptors that are known as opioid receptors, and they come in several flavors. 3 main receptors that are known as mu, delta, and kappa, opioid receptors. And so at the time when people, and so this, this has been a long problem, right? People often ask me, how come we still have to deal with opioid addiction? This has been known for so long. Why is it so difficult? And so at this time, so you have different receptors that can potentially bind opioids such as morphine. And so there was the possibility that one of the receptors was responsible for the positive effects, the analgesia that we still need in the clinic. Mm-hmm. And maybe one of the other types was the bad one, right? Important for addiction.
Which would be convenient.
Right. Yeah. So then if that was true, then what you need to do is to have chemists doing their work to generate novel compounds that are what we call more selective, that will preferentially activate the nice receptors and not those that are causing the side effects. Unfortunately, so the work that Brigitte Kiefer, my PhD mentor, did demonstrate that that's not the case. It's actually the same receptor, which is known as the mu-opioid receptor, that is responsible both for the pain-relieving properties of opioids, but also essentially all the side effects. So the rewarding properties that cause the transition to addiction, the respiratory depression. So one of the reasons why opioids are so dangerous is that not only they are addictive, so many drugs or habits are potentially addictive, but opioids have this particular property that they have receptors in the brainstem, in the respiratory centers. And so most people who die by opioid overdose die by respiratory failure because opioids directly inhibit respiration. And since they're also like sedative, people can fall asleep and essentially stop breathing in their sleep. So that's quite different if you think about cocaine, for example, or psychostimulants. They get you excited. So, and they don't have this effect on the respiratory system. So you can still die from overdose, but it's much less common than with opioids.
I see. So they're sedative, and they can depress the respiratory system. So right. The fact that
that's
you get those
very
two
dangerous.
things, those two things simultaneously, you just fall asleep.
Right. Exactly. And so. So what Brigitte did at the time, she had just cloned the genes that encode the different opioid receptors. And so she made knockout mice, so mice that are completely normal, except that they don't express one of the opioid receptors. So you can think about this as 3 different strains of mice that lack either mu, delta, or kappa opioid receptors. And it turns out that if you give an opioid, whether it's morphine, oxycodone, fentanyl, to the mu-opioid receptor knockout mouse, this mouse is essentially immune. Nothing happens. You don't have pain relief, but you don't have addiction, you don't have respiratory depression, you don't have constipation, nausea, vomiting. All the side effects of opioids are gone. So that's a much bigger problem, right? Because instead of having to work with chemists to make better molecules, now you have the same receptor that generates both the positive and negative effects. So how are you going to tease that apart? And so this paper on this mu-opioid receptor knockout mouse was published in 1996, and we still don't have a solution. So you can see that's really the core of the problem. It's like the same receptor is both good and bad. And compared to other drugs of abuse, the drug that's abused is an FDA-approved drug that's absolutely essential in medicine. No hospital could function without opioids.
Because they are so effective at pain.
And we don't have alternative, right? So if you want to treat moderate to severe pain, there are several other things you can treat. But at some point, most patients will need opioids. That may or may not work. It depends. But it's still the best option for severe pain.
And so, we could ask 2 questions, which might really biologically be one question. Why are opioids so good at generating pain relief and so addictive? Are these mu opioid receptors responsible for both the pain relieving effects and the side effects? Are they in like really important specific parts of the brain? Are they all over? Like what parts of the brain make it so potent as a pain agent and an addictive agent?
Right. So for the pain part, what's really interesting is that this receptor, the opioid receptor, is essentially present all along the pain circuitry. In the primary afferent sensory neurons that detect painful stimuli in the periphery, in the spinal cord neuron that receive and process that information, and in the brain center that create the percept of pain. So if you wanted to start a new project and find a way to treat pain, you would probably pick a receptor that is present at all these spots. And it turns out that that's where this receptor is. And it's a receptor that, in terms of molecular signaling, is inhibitory. So it's going to reduce the ability of pain-sensing neurons to communicate this information to the downstream neuron in the circuit in a way. Mm-hmm. So several things. In terms of neurotransmission, opioid receptors are coupled to different effectors in the cell, in the neuron. And so several things happen when an opioid binds to its receptor. First, these opioid receptors will inhibit calcium channels. So these are ion channels that are very important for neurotransmitter release. Mm-hmm. At the synaptic terminal, when neurons fire action potential, there's going to be an increase in intracellular calcium. And this signal will cause the fusion of synaptic vesicles with the plasma membrane. And so then the neurotransmitters that are inside the synaptic vesicles will be released onto the next cell to sort of activate or inhibit it. And so what opioid receptors do when they're activated is that they're going to diminish the ability of these calcium channels to let calcium in. So in the presence of an opioid, you have less calcium flowing in the synaptic terminal, less transmitter release. So this neuron is less able to communicate pain information to the next neuron in the circuit. Mm-hmm. So that's one effect. And the other effect is through another kind of ion channels, potassium channels. And that's regulating the excitability of the cell. So in that case, opioid receptors, when they bind fentanyl, can open potassium channels. So more potassium flows out. And since this is a positive charge and neurons already have more charges that are positive outside the cell compared to inside, you do what we call a hyperpolarization of the membrane. So now it's going to become more difficult for these pain-sensing neurons to get excited in the first place. So you really have these two effects on the neuron. First, they're less easy to excite. And then even if they're excited, they cannot really communicate this information very well. So you can imagine that if you do that multiple times along the pain circuit, it's going to be quite effective to reduce pain.
Yeah. So the odds of any signal getting through are quite low because you have this inhibitory effect. You've got it at the places in the periphery where pain is first detected. You've got it in sort of the middle relay stations of the spinal cord that connect the periphery to the brain. And then you've got it additionally all the way up in the brain. So there's very little room for something to get through.
So for opioids, even though the receptors are also present in sensory neurons, there's good evidence that most of the analgesia probably comes from action in the brain and spinal cord.
Mm-hmm, mm-hmm. I see. And I want to give people a little bit more of a sense for how pain information is detected and relayed in the absence of a drug. So, let's just say, if a needle or something or a pin pokes me in the arm, obviously, I feel that pain. I know where it is. I can localize it to the part of my arm where I got the pin prick. Can you kind of walk us through on a very, very basic high level, like how is that detected and relayed up to the brain?
Right. So, when you have this discussion, you have to think about the percept that your brain creates. So that what you just talked about is more what we call the sensory discriminative aspect of pain. And so you're able to distinguish all these stimuli. For example, you can know if it's if you were cooking, you put your hand on the hot stove, you get distracted, you can tell it's painful, but it's also burning heat. That's different from if you drop something very heavy on your foot, right? Mm-hmm. You can tell. And so you can tell because we have different kinds of sensory neurons that innervate skin, muscles, bones, viscera, etc., that are called nociceptors. So these are the sensory neurons that are specialized in the detection of stimuli that are intense enough that they can damage our body, okay? Mm-hmm. But these come in many flavors. So some are specialized in the detection of the pinprick. So they have transducers that respond to this type of mechanical stimulation. Others are specialized in detection of thermal stimuli, noxious heat or noxious cold. And so we often think about the system for pain, at least in the periphery, as a label line system, so like a freeway with different lanes, where cables detect a particular type of stimulus because they have the sensors that do that. And then they sort of transfer this information to the spinal cord and to the brain, because based on where the stimulus is on your body and what kind of stimulus it is, you can react differently, right? So itch is not exactly pain. It's also unpleasant. But for example, when you're in presence of a painful stimulus, you have this withdrawal reflex.
Mm-hmm.
And then, if you, again, burn your hand on the hot stove, you have your withdrawal reflex. Then you probably go to the sink, turn on the cold water. If it doesn't get better, you go to the doctor, all these things. If it's a peritogen, so a molecule that causes itch, you don't withdraw. You first reach and you scratch, right? So this shows you that the ability to detect different types of noxious stimuli or peritogen or light touch is because we have these different types of sensory neurons that can help the brain decode what has just happened on our body.
Mm-hmm. And so it sounds like, there's many types of sensory neurons. There's many types of pain sensing neurons. So there's sort of diversity at two levels. And it sounds like, so on the one hand, we can detect painful stimuli as distinct from non-painful stimuli. But then even among the pain sensing neurons, there's different types. So we can distinguish between a burn versus, something that would cause a bruise or that's putting a lot of pressure on the body. And it sounds like an important component of the pain sensing neurons is intensity. Like the stimulus has to be intense enough to trigger them. There's probably other sensory neurons that don't respond to pain, but to respond to innocuous touch and things like this. And they probably have different thresholds of firing in terms of the intensity.
All right. So intensity coding is an interesting question. This happens through both cellular and molecular mechanisms. We have types of sensory neurons that are called low threshold mechanoreceptors. These are the ones that typically detect light touch. And these cannot detect stimuli that are too intense. So in the normal condition, we can talk about what is called tactile allodynia, when touch becomes painful. That's quite interesting mechanism. But normally, these cells are unable to fire when the stimulus is too intense. Nociceptors, it's the opposite. If you don't reach a certain intensity that can damage the body, they don't respond. So there's that. And then you have within a certain cell also intensity coding, whereby some cells fire more when the stimulus is more intense. So you have these two main ways to sort of code intensity, at least in the periphery.
And then, when a painful stimulus is detected, whether it's a high pressure stimulus or a very hot stimulus or something like that, what happens next? Where does it go from the sensory periphery? Does it have to go through the spinal cord and particular pathways in there before going up to the brain?
Yes, it does. And this is well known. If you have a spinal cord injury, of course you lose the ability to move, but you also lose sensation. So things that are painful are like a noxious stimulus on your arm or on your leg. If it's a spinal cord injury at a high level, cervical level, you lose also the sensation. Yeah. So the system is the sensory neurons are unusual. So most neurons in the brain, in the spinal cord, have three main parts. But one part are called dendrites, right? This is where the information comes in. Our neurons listen to other neurons that are located upstream in the circuit. And then they communicate this information using their axon. Sensory neurons for pain are not like this. They're called pseudo-unipolar neurons, and they don't have dendrites. So they don't listen to other neurons. They don't care. The only thing they care about is what happens in the organ that they innervate, whether it's a visceral organ, skin, bone, muscle. And so they're called pseudo-unipolar because they have one axon that leaves the cell body. So the cell bodies are in ganglia that are very close to the spinal cord. They're called either dorsal root ganglia for most of the body, and trigeminal ganglia for the face. So the ganglia are located very close to the spinal cord, and then they have one axon that bifurcates, sending one branch that is very long to anywhere in your body. So you can have, for example, a sensory neuron that is in a ganglion at the lumbar level, and that innervates the tip of your toe. So this sensory neuron axon is very long. And that's where pain information is detected in the periphery. Then through action potential firing, pain information is transferred back to the cell body. But in fact, this axon sends a second collateral, another branch, to the spinal cord, rarely to the brain, but there are some neurons that do that. And so then this information gets to the spinal cord, where many types of neurons will process this information, and a specific class of spinal cord neurons, known as projection neurons of the anterolateral tract, would relay this information to the brain. And at this point, the information is emotionally inert. There's no emotion or percept yet, but something has happened in the periphery, and the brain is going to be aware of this.
Mm-hmm. And there's almost, it's interesting to think about the difference between the detection of pain and the creation in the brain of the conscious perception of being in pain. And sometimes you can almost experience the difference there, in the sense that, if you pay very close attention to your experience when you put your hand on the stove or something, the painful experience often comes with a lag, right? It comes after you sort of detect that something's hot and withdraw your hand. There's almost like a split second that goes by before the pain actually starts. Does that have to do with the fact that it just takes time for the peripheral signals in your skin to get all the way up to the brain?
Right. Well, yes, to some extent. But in that case, in your example, you also have learned that the stove is hot. And so you see it. So there are lots of things that happen here. Maybe we'll talk about the cognitive aspect of pain if we discuss the placebo analgesia aspect. But yes, mostly this difference between the withdrawal reflex and the percept is due to the fact that when sensory neurons that have detected a painful event in the periphery get to the spinal cord, almost immediately through one synapse, they activate motor neurons for a withdrawal reflex, such that you can preserve your body even before the percept is generated. And
so the withdrawal reflex, the signal in the nervous system that gets you to safety or gets you away from harm, it doesn't actually need to go all the way up to the brain.
No, it does not. Yeah. In fact, there are many studies that have been shown where if you use preparations where the communication between the spinal cord and the brain has been inhibited somehow, you still have the withdrawal reflex when you have a noxious stimulus. And so that's important, right? Because you want to remove exposure to the noxious stimulus as soon as possible. And then there's enough time to think about what just happened and make a decision about what's the next move. But first of all, you have to get out of the way. Yeah.
Mm-hmm. So are things segregated in the spinal cord? So for example, obviously if someone has a complete transection of their spinal cord, it's completely cut, nothing can go up or down. You can't move certain parts of your body depending on where the lesion is. And you also can't feel those parts of the body. What if you were to say damage part of the spinal cord? Is one part of it for signals going out to the muscles to move your body and another part for the inputs coming in to sense things, or are things all sort of a mishmash?
In theory, yes, but these things are pretty close. They're close enough that, normally with an injury that would happen, for example, with a trauma, a car accident, you would probably have both types of problems. So the way the system is organized is that the sensory neurons that project to the spinal cord are relatively close from the axons that come down from motor cortex to produce movement. So in our research, right, when it's not an accident, but in our research we can use tricks such as optogenetics, chemogenetics to manipulate specifically neurons that are either part of the sensory or the motor system, and we can tease these things apart. But with a natural injury, these things are pretty close. So
I see. So in principle, the sensory and the motor side, they're functionally and they're anatomically distinct.
Right.
You could, if damage is just right, you can damage one but not the other. But in practice, based on just the raw size of things, if you get a spinal cord injury, you're probably going to take out the sensory and the motor component.
The spinal cord is pretty small. So, and usually when there's an injury of this kind, if it's a severe trauma, spinal cord take a hit, and you can still have selective deficits, but anatomically, these things are pretty close. It's not like, the front part of the brain and the back part of the brain, things like that.
Interesting. So painful stimulus is detected in the periphery on the surface of the skin, say. sensory neurons are doing that. These sensory neurons are different from a lot of other neurons. So instead of listening mostly to other neurons, these neurons are listening to the external world by penetrating a given tissue, and so they're different in that sense. They send their information to the spinal cord, and then it gets up to the brain eventually. We don't need to be fully comprehensive here, but what are some of the major brain regions, the major players in the brain that are involved in processing pain?
Right. So you said, external world, but also inside our body, right? So viscera, for example. Mm-hmm. So we have different sensory neurons that some are projected to the skin. So obviously in that case, it's for the external world mostly. Mm-hmm. But most sensory neurons are going to innervate our own tissues where they can also detect diseases, problems.
I see. So if I have, if I have an upset stomach and there's some kind of inflammation in there, or maybe I've got a bleed internally, there's actually sensory neurons detecting that in a similar fashion to the ones in my skin.
So, the stomach, you could also argue that external world, right? Because you just got some food, like you ate something very spicy.
Yeah.
Your sensory neurons are not going to be very happy about that choice of meal. But that's still the external world. But for example, and this is relevant, for a number of diseases, including cancer. So organs where, that are densely innervated are more likely, for example, to detect the growth of a tumor than other organs that are less well innervated in terms of sensory neuron density. So, you might know that, for example, for certain types of cancer, like pancreatic cancer, one of the issue is that so often diagnosed late because it's not painful early.
Because it's not innervated very much?
Or because, the changes in the organ are not sufficient for these sensory neurons to be engaged.
I see.
So, the pain system in a way is its efficacy is different based on the organs and the type of stimuli.
I see. So pancreatic cancer is diagnosed late typically because people just never have a painful stimulus early on.
Well, never, or it's
Rarely,
at a stage where the tumor can be quite large.
Yeah.
So that has to do with the skin is the opposite, right? So when you have pain that comes from internal organs, often it's like dull, not very well localized, right? On the skin, it's extremely precise. That's because the type of sensory neurons that innervate different organs have different receptive fields that are, smaller or larger. And so our brain gets some information that is more or less precise depending on the body parts and the properties of the sensory neurons.
So these sensory neurons can be bringing information that originates in the outside world, like, something touching my skin. They can also be bringing in sensory information that originates inside, somewhere in my viscera. That stuff is being relayed up to the brain via the spinal cord. And what are some of the major nodes in the brain that then take over?
Right. So, you can think about pain as a multidimensional experience where there are three main components: sensory, emotional, cognitive. So for sensory, that's, where is the stimulus on my body? What is it like? Is it, heat? Is it mechanical stimulation? And so that goes through the sensory part of the thalamus and then the somatosensory cortex. And here there's, a map of our body where these stimuli are represented, and our brain can decode where the noxious event has occurred inside your body, in that area, on the skin, on the hand, on the foot, etc. And so that information is important because the first thing you're going to do after the withdrawal reflex is what's going on. So you have to know where the painful event has been to put your hand under the cold water, put a Band-Aid, deal with it. So that's the sensory discriminative aspect of pain. And then you have the emotional aspect of pain. So that, that's the aspect of pain that's essential for survival. The only reason we care about pain and we deal with it, we go to the doctor, we do what's necessary, is because there's this aversive signal that is generated during perception. So we have to dislike pain to consider, okay, this is an important thing that I don't want to, experience very long. So I'm going to do whatever it takes to make pain go away. And that's happening in different areas of the brain. It's not, overlapping with these other areas that are important for sensing pain. Also the thalamus, but other nuclei that are located less laterally, but more on the midline. and then other cortical areas or subcortical areas, such as the amygdala, cingulate cortex, insular cortex. So there's really like these two main pathways that detect pain sensation or generate pain sensation, and then generate the emotional affective component of pain that's necessary for survival. So people who have mutations, for example, in some sodium channels where they can sense pain, they can generate the negative aspect of pain, so the unpleasantness, they usually don't live long because they have injuries in their body and they don't deal with them because they're not bothersome.
You break your leg and you keep trying to walk on it.
Yeah, I still want to go, watch a movie or whatever. It's okay.
Interesting.
So it's really the unpleasantness of pain that is crucial, and that is the problem. That when pain persists and becomes a disease, this is really where it's really problematic for patients. It's not to know the representation of the painful stimulus in terms of where it is. It's really this aversion that's generated and that prevents them from doing things that they like.
Yeah, so it sounds like the painful information that's getting to the brain. So it's these nociceptive neurons have detected that pain is there in some form or another, somewhere in the body. That information gets up to the brain. The brain then has to solve a number of problems. The one is, as you mentioned, where is the pain coming from? And so that's where the somatosensory cortex comes in, and the brain sort of maps, using its map of the body up in the somatosensory cortex, where the pain actually is on the body. There's also the emotional component. The pain needs to be perceived as aversive in order for the whole organism to learn sort of to dislike it and avoid it in the future. Where are some of the brain regions that are responsible for that aversiveness?
Well, so one important site is the amygdala. So that's a region that's important for emotions in general, positive emotions, negative emotions, so things we like, things we dislike. And so there are a number of studies that show that for this particular pathway where neurons that are in the spinal cord project this pain information to a small structure that is the parabrachial nucleus, then relay this information to the amygdala. And so there are case reports that show that if you have a lesion in this pathway or in the amygdala, the sensory discriminative aspect of pain can be conserved. So people can still detect stimuli, but they are no longer bothersome. And so we've shown in rodents too that if you artificially inhibit cells in the amygdala, you can disassociate these two aspects of pain experience, the sensory aspect and the emotional aspect.
So the amygdala really does seem to be involved in the emotional aspect here.
It is, yeah.
And then, of course, layered on top of that, there's this sort of cognitive or higher level processing that can happen. So to sort of connect this to everyday life, there's actually many examples that we probably all experience where something is painful or at least uncomfortable, and it's naturally experienced that way, but nonetheless we do it or even keep doing it or even learn to enjoy it. So maybe I decide I want to go to the gym tomorrow and start working out. I start lifting weights. My muscles burn during the experience. My muscles ache the next day. These aren't pleasant experiences. I don't like them, but actually I can make myself keep doing this anyway. And maybe over time, I actually come to enjoy the sensation in some sense. So how does the cognitive layer start to fit in here?
Yeah. These are great examples. So in systems that are less developed, for example, some invertebrates, the priority of the pain system remains to avoid noxious stimuli that can damage your body or kill you. And so in these systems, usually noxious stimuli equals escape. But then as animals evolve and become more intelligent, we all know sometimes we have bigger plans. There are things we need to get done. And so we need to have the ability to calculate if it's more important to prioritize avoiding pain and not do the other things that we had planned, or if the pain is not too severe, too serious, we can decide to ignore pain. So, you're a tennis player, you've been practicing your entire life, you're finally in the final of the US Open, you have a light sprained ankle, in the first set, your cortex is going to tell your amygdala, okay, keep quiet because I know what this is. I've had all the sprained ankles. I've been working towards this goal for a long time. I can deal with it. I'm going to decide to continue, playing tennis. And so it's crucial to to achieve long-term goals that can be very important goals for survival as well, to be able to make this calculation based on our previous history. If we expect this pain to be something transient that's not, severe, or if it's something that's really important that hasn't been encountered before, where we're sort of like, okay, well, this seems really bad. I've never felt that before. Probably should go to the doctor. So yeah, this cognitive aspect is like the dimension that's very important, becomes prevalent, I would argue, in humans.
And, obviously that's very important, especially for organisms like ourselves that can engage in long-term planning. We can prioritize how much we want to avoid some temporary pain right now in order to achieve some larger goal that will pay off later. Another thing I want to think about here, get people thinking about is, so we've talked about these different components of pain. There's the sensory side, there's the emotional side, the actual experience that is unpleasant, and then there's the cognitive dimension here, the thing that can tie into, goals and planning and, higher order stuff like that. There's also maybe another distinction we could make between different types of pain experiences. So people might make a distinction between physical pain and emotional pain. So they're both going to result in an emotional pain experience that's unpleasant and aversive. But one of them, if I smash my finger with a hammer, there's a clear physical cause that's related to tissue damage. But if, say, a family member dies or a pet dies, there's no tissue damage there, no potential tissue damage to my body. And yet we talk about it as being a very painful stimulus. Is that a valid distinction to make? And do those types of emotional experiences tap into the same pain circuitry centrally?
It's a very valid, or it's an important question. So, if you're a researcher in a pain field, the second case where there's no physical damage is not really pain, right? We use this term pain like, I bet on this team and, I lost. This is painful. But no, it's not painful. Nociceptors have not been activated. The signal that's been generated in the brain is not really pain. It's something that we perceive as an unpleasant experience. But, there's no actual or potential tissue damage, which is really the definition of pain. So pain as a definition is like unpleasant experience with sensory and emotional components, but it has to be associated with either frank actual tissue damage or the potential for such damage. So, but there's overlap, right? In the brain regions that are active when we have an unpleasant experience, whether it is actual physical pain or something that is, as you said, emotionally painful, the cingulate cortex, number of other regions are activated. So there are areas in the brain, in the periphery, that are a little bit more pain specific. We call them nociceptive neurons. And there are other areas in the brain where neurons do all sorts of things. So they can get engaged during pain. That's unpleasant, but other things that are unpleasant or, pleasurable.
Now, so far we've sort of talked about the natural way to think about information flow, which is sensory detection in the periphery, relayed through the spinal cord up to the brain, further processing happens. Can things move in the opposite direction? And I want to ask you that in the context of something like psychosomatic pain. So can I form beliefs in my cerebral cortex that actually feed all the way back down to my nociceptors in the periphery to create a pain percept in the periphery that's entirely internal in origin?
That would be less common, because that would mean that you have, your brain has created a pain percept that is very specific and associated with a particular body part that would have simulated, it can happen, but that would have simulated exactly activation of a discrete set of sensory neurons that only innervate, the dorsal surface of the arm, and that this is, perceived as painful. The top-down organization of the pain system is more as a modulation system, whereby, I can give you the example of the fight or flight response where, a firefighter goes in a house on fire, saves a baby, super happy, no pain, reports to the hospital, very severe burn pain that should be excruciating. So what has happened in that case is that there are neurons in the brain that have activated this descending pain modulation circuit, so that, starts in the cortex, but goes through regions such as the periaqueductal gray and then rostral ventromedial medulla that then projects to the spinal cord. So we have brain neurons that can regulate how much nociceptive information is going to transmit from sensory neurons or spinal cord neurons back up to the brain. So the pain system really has an ascending component and a descending component, and many connections and interactions between these two systems. When you have, for example, phantom limb pain or types of pain like this, when the sensory neurons are not actually activated by a noxious stimulus.
Yeah, like I guess in the case of phantom limb, they're not even there anymore.
That's right. They're not even there, but there's a part of the pain circuit that's still there. So, for example, part of the nerve that can still be there, or the corresponding spinal cord neurons postsynaptically. And so when there's activity in the nerve or in these spinal cord neurons for our brain, it's as if it was actually coming from the terminals that are in the skin or in the organ. So the brain cannot sort of erase this percept that the arm or the leg is still there. And so in that case, you can have types of pain that are perceived when there's aberrant activity in the nervous system, and the brain just doesn't have a way to figure out that this is not an actual noxious stimulus, but it's just activity in the circuit.
Yeah. From the somatosensory cortex's perspective, the same part of the body map gets activated.
Yeah, and so this could happen many places in the spinal cord or brain, right? If you have aberrant activity for your somatosensory cortex or other parts of the cortex, it's going to look the same.
And we've mentioned exogenous opioids so far. They're very effective and very addictive because of their ability to inhibit neurons via the mu opioid receptor. And part of the reason for that is this receptor is found sort of at all stages of this pain circuitry from the sensory periphery all the way up into the brain. I want to ask you a little bit more about endogenous opioids, things like endorphins and enkephalins that you briefly touched on. What are some of the normal, natural situations in which the brain releases these endogenous opioids?
All right, that's a great question. Can I just tell you one more thing about I think you had a question at the beginning that was about, pain relief versus addiction. And so I said, opioid receptors are inhibitory. And so this is why they reduce pain. So if you think about that molecular logic, you might think, they should reduce addiction too, because they're in, addiction neuron, therefore they inhibit addiction neuron. They shouldn't be addictive. So just to clarify now this works, so there's another area of the brain that's called the ventral tegmental area that has dopamine neurons. So dopamine is one of the transmitters that make us feel good when it's released. What happens in that case is that opioid receptors are not in the dopamine cells. They're inhibitory neurons that control dopamine cells. So this is
you're
what
actually
we
disinhibiting the dopamine
exactly.
neurons.
So when opioids are on board, they're going to turn off the inhibition of the dopaminergic neurons. And so this is why mostly in the pain system, opioid receptors are inhibitory, but they are in the pain cells, not in the cells that inhibit the pain cells. But for the reward system, unfortunately, they're not on the cells that are rewarding themselves, but on cells that control the rewarding cells.
I see. So if you go to the doctor and you get an opioid pain medication because you actually have a physical injury, the pain will go away or be diminished because you're inhibiting the neurons that actually generate the pain signal, but it will feel good. You'll feel euphoric and so forth because you're actually disinhibiting or activating effectively the reward neurons.
Right. And to pick up on your question with, endogenous opioids, what's interesting is that we are not addicted to our endogenous opioid peptides, right? So what's the difference? And so, these peptides are labile. They're released by the cells, but they sort of float around a little bit and then they're degraded. In contrast, and they're peptides, so they cannot really cross membranes well, right? They bind the opioid receptor, they don't go inside the cell. In contrast, something like morphine is going to be in your body for hours. And so there are several things that happen, right? They can activate opioid receptors where at the cellular level, the peptides cannot go. For example, there's data now showing that there are intracellular receptors that traffic in cells that can be activated by exogenous opioids, and peptides normally do that. Don't do that, and they're around for hours. So instead of being like a fine signal that sort of fine-tune neurotransmission, which is what peptides do.
Yeah.
These drugs massively activate opioid receptors for hours. So they have very different consequences in terms of how neurons function.
Yeah. So the endogenous opioids are spatiotemporally precise. They're sort of used where and when they're needed. Whereas, if you take an opioid pill, it's going into systemic circulation. It's sort of hitting all the receptors everywhere at once. It's sticking around longer. It sounds like there's an analogy there with cannabinoids, right? Endogenous cannabinoids are, they don't last very long. They're used on a synapse to synapse basis, so they're very localized spatially. But if you take an exogenous opioid or an exogenous cannabinoid like THC, it's sort of hitting all of the receptors at once everywhere.
Very similar for, most drugs that act on these categories of receptors, like the opioid receptors, are called G protein-coupled receptors. That's what they're useful to treat disease because, you can get it once or twice a day. And for a relatively long time, through signaling of that particular receptor, you can, resolve your high blood pressure problem or whatever problems you might have.
Mm-hmm. Yeah. So going back to this notion that, right, we've got multiple sort of layers to pain. There's the sensory side where you first detect the pain. There's the emotional side, the actual subjective experience of pain, the aversiveness or unpleasantness of it. And then there's these cognitive modulations of that experience. One of the things that you've studied recently and done some interesting work to work out the circuitry behind this has to do with the placebo effect, which is pretty well known, especially as it relates to pain. But nonetheless, I want to start out very basic here. So what is the placebo effect as it relates to pain?
Right. So placebo effect is something that we know very well in the pain field for several reasons. But one of them is that we just talked about opioids quite a bit. One of the reasons why opioids are still used clinically is that we don't have good alternatives. One of the reasons why this happened is that many drugs have entered clinical trials, and a number of them have failed not because they didn't have some level of pain relief, but because they didn't beat the placebo group. So when you do a clinical trial to get FDA approval, you have to demonstrate that your treatment is useful, that it's better than the reference treatment, but you also need a control. And so in the pain field, it's known that people who are in the placebo arm often have a very strong response. So they have a level of analgesia that is high, that makes it hard for the new drug to demonstrate efficacy beyond the placebo effect. And so it's a reduction in pain perception that results from the expectation that pain is going to be reduced or go away in the absence of any actual treatment.
Mm-hmm. And do we understand what the physiological basis for that effect is? So, for example, when people believe that the sugar pill is going to relieve their pain, do they have a release of endogenous opioids or something like that?
Right. So there's been, many studies on what is placebo and how it works. Much of it has been done through human imaging studies. That's in part because we'll get to that, but placebo is not easy to model in mice because it's through verbal suggestion where your doctor is going to tell you, I'm going to make this prescription. It's going to be great for you. It works in everybody. And then you have this placebo effect. It's hard to model this in the mouse. But so, yeah, what happens in that case is that you're forming this expectation. And sorry, going back to your question, that was...
Oh, yeah, it's just like, so the placebo effect. So I'm told by a doctor that this pill will make me better. It's a sugar pill. I don't know that, but I believe it's going to make me better. And then I actually experience pain relief. So, it's a real effect. Is that due to endogenous opioids or something like that?
Right. It is. Yeah. So these are the few things we know. So through the human imaging studies, we know the parts of the brain that are activated during the placebo effect. And the other thing we know from studies by John Levin and Howard Fields in the 80s is that if you give naloxone to people, you're going to reduce the magnitude of the placebo effect. So naloxone,
it's an opioid blocker?
Yeah, an opioid receptor antagonist. So the same molecule that's given in DR to save people's life when they overdose. So this blocks all opioid receptors. And so the fact that you have a reduction, sometimes elimination of the placebo effect in the presence of naloxone tells you that it is indeed a release of endogenous opioid peptides that is, the molecular mechanism by which the placebo analgesia is caused. So somewhere in our brain, when we have an expectation that pain is going to get better, some neurons can release endogenous opioids, and that elevates pain threshold. Pain is more, acceptable or seems to be less intense.
So the opioid system is involved in the placebo effect. If you use an opioid receptor blocker, can you prevent the placebo effect from happening?
Yeah, that's what happens. Yeah. So in these studies, sometimes there are other mediators that are involved, but usually in most studies there's a strong reduction in the placebo pain relief when opioid receptors are blocked.
Yeah. And so it sounds like, in human beings, we can kind of figure out with imaging what parts of the brain are involved in the placebo effect. We can figure out that the opioid system is involved, that you could block it with an opioid blocker, say. But we can't really get in and understand the circuitry in fine detail. To do that, we'd have to do invasive experiments in animals. And as you mentioned a few moments ago, it's really hard to study the placebo effect in mice because how do you build that expectation into them? But nonetheless, you guys seem to have come up with a way of doing this.
Yeah, so one thing mice are pretty good at is associative learning. And so what we did was to design an assay where we have two chambers that are next to each other with a small door in between. And so if you put a mouse in that environment, the mouse can freely move between the two chambers. And so what we have is that in that assay are also distinct visual cues in the two chambers. So black and white lines that are either horizontal or vertical. And mice are very good at distinguishing these two things. And so then we are going to condition the mice. So it's a condition-based placebo analgesia model. The way we're going to do this is that we have the ability in this test to change the temperature of the floor. Okay. So we start, it's a 7-day test, where we start by 3 days of habituation, where the floor is at 30 Celsius degrees, which is very comfortable. Mice like it. And so they are naturally, like us, driven to explore. So they're going to spend their time moving around, transitioning between the two chambers, and getting familiar with the environment. And we do that for 3 days. And then on the 4th day, so from day 4 to 6, we're going to trick them. The first chamber where we place them at the beginning of the assay, we're going to set the temperature of the floor at a noxious temperature, 48 Celsius degrees. So when we put mice on that floor, they now know the environment. They will explore to try to escape heat pain. And they will learn that if they move to the other chamber that is still at 30 Celsius degrees, pain will go away. So they will experience pain relief by transitioning from the first chamber to the second chamber.
I see. So you're teaching the mice, you've got two chambers, they look a certain way, they smell a certain way. They start out at the same temperature, but then after a few days, you make one of the chambers too hot to handle for the mice. They don't like it. And they learn, okay, if I go into this one, bad, too hot. And if I go in this one, comfortable. So you just train them to discriminate these two locations based on temperature.
And we start the test by putting them on the hot side. So they have no choice. They have to find a solution to their pain problem. And so the treatment that they find is to go in the other chamber.
And so you can then directly measure and quantify how much they're expecting the other side to be hot, how much they don't like it by how much time they spend in the other chamber.
Exactly. And so they're really good at this. After just 1 day, or certainly after 3 days, you see that they spend almost all their time in a chamber that's not hot, and their latency to transition from the hot chamber to the chamber that is at 30 Celsius degrees is getting shorter and shorter. So they really learn the task well. And so here we think we have, after 6 days, recreated the expectation of pain relief. So then the other component that we need to test is there actually pain relief? Do they experience less pain? And so for that, on day 7, we're going to trick them one more time. Now we're going to set both floors at 48 Celsius degrees. So they've just learned that where they start, they just have to move to the other side and experience pain relief. In that case, they go on the other side, it's still hot. And it's the exact same stimulus intensity. So they should perceive the same pain on both sides. However, when we measure pain with our assays, so we look at licking of the paws and movements that suggest that mice are perceiving pain and want to make pain stop, rearing, jumping to escape, what we see is that mice still show much less pain behaviors on the side that is associated for them with pain relief, even though it's the exact same
stimulus intensity, same temperature, but they're showing less avoidance behaviors or less vigorous avoidance behaviors, presumably because they have this association that's baked in.
Yeah, exactly. And going back to the opioid part, if we give them naloxone on day 7 before testing them, we lose this analgesic effect that's related to the expectation of pain relief.
I see. So you've created a true expectation. Then you can actually block the effect. You can give an opioid antagonist, and now when they go to the second side that used to be comfortable, it's now also noxious like the first side. They respond with pain-related behaviors, avoidance behaviors that are just like the other side.
Right. So we have 2 groups of mice. So the mice we just talked about and their siblings that go through the same 7-day protocol, but they don't have the day 4 to 6 when they were conditioned. And so we compare pain behaviors of mice that have been conditioned to expect pain relief on the right side of the assay to those that have been going through the same session but without the conditioning. And if we compare behaviors for avoiding pain perception in these 2 groups of mice, we see a strong reduction in those that have been conditioned, even though they are physically exposed to the exact same noxious stimulus.
So you can train mice to do all of this stuff. They learn very quickly to avoid the painful side, engage in certain behaviors after they experience a painful stimulus if they go to that side. And there seems to be this opioid system dependent expectation effect akin to the placebo effect involved. How do you actually then look inside the brain at what neurons are doing while all of this is going on?
All right. So that was the first phase, right? It's always, for us, sometimes the most difficult part is how can we model the human phenomenon? So at that point, we felt like we had a good model to study the system. And then, reading the human literature, we found that there was evidence that one part of the brain, particularly the cingulate cortex, was active during the placebo analgesia task. So we used a number of techniques, but one of them is to use miniature endoscopes that can collect fluorescent signals inside the brain. It's called a miniscope. It's been invented by Mark Schnitzer at Stanford.
So you literally mean like a miniature microscope that is hooked up to the animal's head.
Yeah. So it's tiny. It's about 3 grams. And so you can mount this on the mouse head like a hat. And the mouse, because it's slide, mice behave normally. They function completely normally. And so what you can do with this is that you can then use viruses to express in certain neurons that you want to study a calcium indicator. And so when neurons fire action potential, the intracellular concentration of calcium is going to rise, and that's going to change the fluorescent signal that you can then record at the single cell level with this microendoscope that we call a miniscope.
I see. So you can engineer mice such that specific neurons in the brain, in the amygdala, in the cortex somewhere, basically wherever you want, you can make those neurons literally light up if they are sending out nerve impulses or action potentials. And then because you've got this endoscope, this miniature microscope hooked up to the animal's brain, you can actually record when those neurons light up. And so then you can put such animals into the chambers like you just described a few moments ago, and you can watch to see what kind of activity these neurons display as the animals are doing this pain-related learning.
Exactly. So there are other ways to record activity of neurons, but sometimes you have to anesthetize the animals, or there are other aspects of the methodology that do not really permit the type of things that we want to do, which is in molecularly defined types of neurons in a freely behaving animal, what happens when they perform the task.
Yeah. So a strength of this way of recording is that you might not get signals that are as precise or temporally precise as say an electrode, if you're measuring with electrodes, but you have the advantage that you can record from molecularly defined groups of neurons, particular parts of the brain, and the animals can walk around and behave just as they do in normal conditions.
Exactly. And so when we use that technique in our placebo analgesia task, what we found is that as mice are learning the task, that they're moving from chamber on the left to chamber on the right, from heat to comfort, just before they cross and they get on the other side, these cells in the cingulate cortex light up. But not all cells. We found that it's a particular subset of cells in the cingulate cortex that light up. And what's really surprising in that study is that it projects to an area of the brain that's not known as being one of the major center for pain modulation, a region in the pons that's the pontine nucleus.
And so, before going further with these results in terms of what you guys found, so the cingulate cortex, what do we know about it? What is it sort of known for doing before you even went into the study?
It's a very important region for pain perception, and particularly for the affective and cognitive aspects of pain perception. To the extent to which there's actually, it's still happening a little bit, but it used to be a major procedure whereby you could treat intractable pain through surgery by doing something that we call a cingulotomy, where you will lesion the axons of these cingulate cortex neurons that leave the cortex. And that's a way by which you can remove some of the emotional, unpleasantness of pain.
Wow. Under what context would that happen?
It seems a little barbaric, but pain is so difficult to live with that for some patients it's been maybe a little less, but it's a surgery that can still be performed, where you can reduce the affect that is associated with pain experience, which again is really what's problematic for patients who have severe chronic pain.
Well, so if you have severe enough chronic pain and you can't help it through drugs or other means, it's almost like epilepsy. Like there's severe forms of epilepsy which you just can't treat them other ways, so they end up going in and just slicing part of the brain, and that's what actually helps.
Yeah, so that's cutting out the axons of these cells and preventing them from communicating this affective signal. Or generating this affective signal.
Yep. So the animals are experiencing pain if they walk into one side of the chamber. There's an emotional component to that, obviously. They're learning, right? So they have to think, I guess, and do this. So there's a cognitive component. For those reasons, I imagine, because you already knew the cingulate cortex was implicated in these types of things, that's probably why you pointed your microscope there. But then the surprising thing happens that you mentioned briefly a moment ago, where the neurons that were lighting up the most were projecting to a region called the pons, which you probably didn't expect to see.
No, we did not. So, the simplest way by which we could explain that cognition and, for example, a positive expectation about pain, that pain is going to be relieved, would work is, for example, the cingulate cortex that we know is reciprocally connected with the amygdala would send a signal to the amygdala to decrease activity in the amygdala to reduce the aversiveness of pain. There are a number of other regions that we were expecting, medial thalamus, some parts of the striatum, but that's not at all what we found. So this main projection to the pontine nucleus was really intriguing because the only output of the pontine nucleus is the cerebellum. And the cerebellum in, pain neurobiology has always been a little mysterious. It's not easy to study. It's much more studied in the movement field, where we know it's essential for motor coordination and motor learning. In the pain field, it's often seen as activated in human imaging studies, but there's this issue that, of course, when you're presented with the noxious stimulus, you withdraw from it.
You want to move away.
Always involves movement, right?
Yeah.
And so perhaps this signal in cerebellum that is seen during these experimental studies of pain is due to movement, or even just to the intent to move. What we find in our study is that the part of the cerebellum to which the pontine nucleus that receives this expectation signal from the cingulate cortex projecting cerebellum is not the motor areas. There are particular areas of the cerebellum that are more related to cognition, and this is where this signal is going.
And so classically, in textbooks and things, as you mentioned, the cerebellum is associated with movement and motor coordination and motor learning. So historically, it's really been thought of as a motor region, not as a cognitive region. But based on this work and based on some other work that's been coming out, it seems like the cerebellum's reputation is changing, and it is in fact involved in various cognitive operations.
Very much, yeah. So like you said, it's been studied in the context of movement and partly motor learning. Now there's evidence that the cerebellum learns many other things, right? So there are other publications that show, for example, that some types of neurons in the cerebellum known as granule cells can encode the rewarding aspect of a stimulus, such as in a study when mice lick sucrose, which they like, there are some cells in cerebellum that are turned on. So it looks like the cerebellum is important to learn about pain and some other things, and do many more things beyond motor control. So we're quite excited now in understanding what the cerebellum does with that information and how it's able to reduce pain. And so we've started some studies that are not part of the paper that just came out, where we find that, in fact, the cerebellum is very well placed to control pain, because in terms of outputs, you have cerebellar nuclei that are well connected to important places of the pain circuitry, notably the descending pain control circuit, the periaqueductal gray, but also projections back to thalamus, to cingulate cortex. There are even Purkinje cells that project to the parabrachial area. So this first region in the brain that gets information from the spinal cord before sending it to the amygdala. So when we look at the connectivity of the cerebellum, it actually makes a lot of sense that it can modulate pain. And there's this also intriguing association between movement and pain. If you think about when you sprain your ankle or you have low back pain, you change your posture. So that's in a way also about expectation, anticipation. You're going to start positioning your movement in a certain way that will reduce the experience of pain by limiting, activation of sensory neurons that cause pain. So it makes a lot of sense that the cerebellum is actually an important part of the pain circuitry. And so we want to push for this notion that maybe textbooks that show these ascending circuits that involve thalamus, amygdala, and then different cortical areas, often the cerebellum is not on these cartoons, and perhaps it should be.
And so what are the implications of this work for humans in terms of how we administer and think about placebo effects?
Right. There are several applications. One first is that's not an application. I mean, it's already an application, in fact. It's cognitive behavioral therapy. So you have three ways to treat pain: drugs, neurostimulation, or behavioral therapy. So there's something called cognitive behavioral therapy, where patients can, learn to live with their pain, be more optimistic about the future. And that's sort of retraining their brain to think differently about their painful condition. And we think that perhaps the circuit that's activated is, has to do with cingulate cortex and cerebellum in that case. So perhaps it's already in use. What we would like to explore is if we can use these two other techniques, drugs or neurostimulation, to artificially recruit these circuits in the absence of conditioning or placebo, just to relieve pain. And so what we show in the study is that, well, first of all, if we turn off this pathway during the placebo anesthesia task, mice do no longer have the pain relief. But more importantly, in mice that are naive and have not been conditioned, if you artificially turn on this circuit, this elevates pain threshold, so less pain perception in mice. So if this is true in humans as well, you can try to engage this circuit either by using transcriptomics, for example, to figure out what's in these cells that we could target to activate this pathway, or use neurostimulation, either in an invasive way or transcranial magnetic stimulation, to generate activity in this circuit and relieve pain.
Interesting. And so, like, how do you think about too? Like, you mentioned earlier, the gold standard for whether drugs get approved is, do they do better than placebo in human clinical trials? Sometimes they don't, sometimes they do, oftentimes they don't. But when a drug doesn't do better than placebo, but at the same time, the placebo effect is real and it's quite strong, and we're starting to uncover the circuit basis of it. I don't know. Is it possible that there are drugs that are sort of working at a similar magnitude as the placebo effect, but they are nonetheless going through a different mechanism, and so they're not actually just, they're actually doing something? You know what I'm saying?
Of course, yeah. That's a great question, and one of my take-home messages when I give these talks is that perhaps we have to reconsider what's really important to have a new pain medication approved, because the people who are in the placebo group do get pain relief. So if, like you say, there's a synergy between the placebo effect that happens and the actual mechanism of action of the drug that makes it useful for people, maybe this is an important consideration and not just a statistical comparison to the placebo group, because the placebo is, we know now, there's a neural circuit that does it. There are molecules that are released, endogenous opioids. And opioids, we know exogenous opioids are very potent analgesics, right? So it's placing on the field a very high bar that the most potent drug that we have an equivalent. Our endogenous peptides are being released, and that's the thing that you try to beat with the new drug.
Yeah, the bar is that you have to do better than one of the most potent endogenous drugs, for lack of a better term, that's inside of us already.
Yeah, or pain modulation system that is there since millions of years in terms of evolution. This is what we use when we do yoga or marathon, and we have a decent pain control. So it's a pretty high bar to beat, and the placebo effects are strong.
Yeah. And so there's also a question here of variability. I don't know too much about this, but my understanding is there's variability in the placebo effect, meaning some people are very prone to strong placebo effects and other people much less so. In the human literature, is that the case? Or in your mice, in the experiments that you described a few moments ago, is there variability from mouse to mouse in the strength of the expectation?
There is, yes, absolutely, like in humans. And that's, this cognitive aspect of pain is also what explains pain subjectivity. Based on our experience, based on the context of where the noxious event occurs, we're going to have a different perception of pain, and that's something that we're really interested in studying. It's not easy in terms of methods because you would, for example, you would want to compare two groups of subjects or two groups of mice and put these mice or humans in the two different groups that are placebo responders and placebo non-responders when you start the study, but you don't know yet, right? So in our mice, we can do the miniscope implants to look at their activity patterns, but we don't know which are the ones that are going to display a very strong effect and those that will not. But we can now, when we have more subjects, we can start doing these correlation analyses and ask, if we go even in the details of the molecular content of these cells, what is different in that mouse that had a very strong placebo response in terms of the amount of endogenous opioids released, the molecules that are present in these cells, compared to the mouse in which the pessimistic mouse, that didn't have a placebo effect. So you really want to understand here what are the molecular cellular substrates that explain pain subjectivity, or why we perceive pain differently depending on the context.
Yeah. And what's kind of interesting to think about too is like if I think about a clinical trial design and I think about the participants in that trial, being a participant in a clinical trial, it's sort of inherently maximizing the odds of a strong placebo effect because, I have to go to a hospital, doctors are talking to me. It's a very formal, very fancy setup. And so you sort of naturally are going to build up virtually everyone's expectations. But then you run into this problem of you might actually sort of mask genuine effects of therapies that simply aren't stronger than the placebo effect, but are nonetheless doing something.
Exactly, yeah, and that's my point. So if you take another viewpoint, which is the patient's viewpoint, where I just want to be better, right? I want to have less pain. Those who have been in a control group and got the placebo lived a better life at the time they were doing the trial, right? Because their pain was reduced. So maybe this is what is important. And if obviously we would want the drug, molecular mechanism of action to add something to this. But again, it's a pretty high bar maybe to try to beat endogenous opioids. So far, it hasn't worked very well.
And so what kinds of studies are you guys doing now or doing next that are related to this work?
So related to this work, we want to understand how it works, so how this pathway is able to reduce pain. So we're looking at the cerebellum, how the cerebellum is connected to the rest of the brain. This is the placebo effect. We're also quite interested in the opposite, which is known as the nocebo effect, where some subjects can develop something that's called as pain catastrophizing, where pain is perceived as worse than it should be based on the injury or the disease or the actual stimulus. And so we have some. So we were just talking about subjectivity and inter-individual differences. We have primary studies where if we start, for example, with 10 mice, and we will briefly expose to the hard floor every day, right? So that they get used to it, but they don't like it, right? So at some point, you put them on there, and at some point it's enough. They jump and they escape, and you do that every day. And so every day the latency for the jump response is lowered. After a few days, you just get it. You put them on it. They're like, okay, I don't want to be there. I just jump. And then after a few days of conditioning like this, we are going to put them back in the exact same context on the same plate, but the plate is no longer turned on. So it's at comfortable temperature.
Mm-hmm.
And you have a certain proportion of mice, between 1 and 3 mice, depending on the experiments, that will still jump, even though there's no reason to jump. So they're very pessimistic. They have a very negative expectation about this based on the context, while it's not that bad on that day. And so what's different in the brain of that mouse compared to the other mice that have made the right decision? Because in the wild, this mouse, by making the wrong move, can also be eaten, right? When you're in pain, you have to hide, you have to protect yourself. So this decision-making is really important, as we discussed at the beginning. And so that, these are the two things: placebo, nocebo, how we make decisions. This is a part of pain that's, at least in terms of basic science and, cellular molecular mechanism, is a little bit understudied compared to all the work that's been done in the peripheral nervous system. Mm-hmm. So, that's one area that's really interesting for us. And we're interested in phenomena in humans that are causing strong pain relief and that we think we can study. So another example of projects we have that's related is distraction. Don't know if you have kids, but anybody who has kids, your kid has to go to the dentist, you're going to give them your phone or the iPad or whatever, and dental pain no longer exists. Right, so distraction is a very potent way to generate analgesia. Same thing. Can we model this in mice? What are the circuits that produce analgesia from distraction? And along the lines of what we do with placebo, when we find the cells, then is there a way artificially to take advantage of that circuitry that exists in our body to find another way to treat pain?
Well, this has been fascinating, Gregory. Very interesting stuff. Is there anything you want to reiterate that we discussed, or any sort of final thoughts you want to leave people with that has to do with pain and pain perception and how we experience pain?
Maybe pain treatment, because I think that's, with the opioid epidemic and many, many people being in pain, this is really a priority. Maybe this notion that ideally we want to have, as many new painkillers as possible. So some that work in the periphery, but I think it's really important to study these other aspects of pain, the emotional aspect of pain, the other cognitive aspect of pain, so that we have different treatment options and ideally things that we can combine. If you think about other fields of medicine, infectious disease, HIV, metabolic disease, diabetes, cardiovascular disease, high blood pressure, cancer, many patients get clinical benefits for having prescribed multiple drugs that act on different aspects of the disease. So I think one thing that we could try to do more in the pain field is to have drugs that we can combine that act on sensory system, on emotional system, on cognitive system, to overall reduce the experience of pain. And that, I think, could be really useful for patients.
Excellent. Well, yeah, again, this was fascinating, and I thank you for your time.
Thanks so much, Nick. Take care.